Okay, starting. Okay, Oded, we're going to share. You can start sharing your slide now. Oded. Okay. Is he on? Okay, Oded, can you hear us?
Yes.
Okay, go ahead and share your slide. Yeah, okay. And share your, make it in slideshow mode. Put it in slideshow mode and then share it. Yeah, we're perfect.
Yeah, but it's not showing up
here. Okay, now I'm sharing. I have to make him presenter.
He's on there twice. Why is he on twice?
Okay. Share your slides. Oh. Okay. All right. Go ahead and share your slides, Oded. Okay. Sharing the slide.
He was on there twice for some
I
reason.
think he seems to have disappeared. Oops. Okay, our speaker just disappeared.
No, there it is.
There. Where?
Oh, no, that's Chris. I'm sorry.
I think he's dialing back on.
Oh, dear.
Oh, there you are. Here he is. Oded, do you want to share your slides? Can you hear us? Okay.
There we go.
Oops.
Okay, now it's better.
There you go.
Okay.
Excellent.
Yes.
Okay. Go ahead and share your. Yes. Share window. Share window.
Share a window.
Mm-hmm.
And now open the slides.
Perfect.
Okay. Put it in slideshow mode. Yes.
There we go.
Okay.
Nancy, can you hear me?
Yes.
Okay.
Yes. Good morning, everyone. Welcome to day 2. Today, we're going to start off with 2 plenary talks on the other kind of photosynthesis, not chlorophyll-based photosynthesis, but retinal-based photosynthesis. We're lucky to have 2 of the world experts on that with us. Our first talk will be given by Oded Beja from Technion in Israel. He's giving this remotely. So his title is How Diverse is Rhodopsin-like Proton Pumping? So Oded, take it away.
Stage is mine. Okay. Okay, so the first few slides are going to be, just a brief introduction. So first of all, there are 2 kinds of rhodopsin families. First of all, animal rhodopsins. You are using those rhodopsins in your eyes and actually see the slideshow with them. But apparently, there's another big family of rhodopsins in microbes. So what you see at the top are the animal rhodopsins or visual rhodopsins. And I'm going to talk about the microbial rhodopsins and different discoveries in the last, I would say, 30, 40 years in one show. Okay, so my lab is mainly interested in marine microbes. And I'm going to describe to you the problems we face. I guess those are a bit bigger problems because basically we cannot culture whatever we want to study. So if this shows you microbes in the marine environment, then if we are lucky, perhaps we can culture one of those microbes. And we have now estimates that about 99% of those microbes are uncultured. And those microbes are doing important things like photosynthesis, CO2 fixations, and other stuff. And the problem is, how do we study them in the lab if we cannot culture them? So one let's say, approach is doing metagenomics. And this is what my lab is doing. So we basically go to a certain environment, in my case, the marine environment, and we collect the entire population as a whole. We can do size fractionation, but basically we collect whatever we want, we extract the DNA, and then we have several methods. One of the methods, this is the initial method that was devised more than 26 years ago, is looking for entire chromosomes, cloning them into E. coli. We have special vectors, and we can do it, and then massive sequencing of those DNA. Another approach, or another two approach, is doing this kind of metagenomics, going to a certain environment, collect DNA, and then chop it to small pieces, and then clone it into E. coli, and then massive sequencing. And lately, most people are doing the different methods of sequencing now, Illumina and other, sequencing directly from the environment or after a small process without the problem of cloning into E. coli. And actually, different labs are using different methods. Most of the labs nowadays, also because of the price, are doing direct sequencing of the DNA they extract from the environment. I'm going to show you a few discoveries using metagenomics, and then you'll see that we are now in a better shape to understand microbial rhodopsins. So this, more than 20 years ago, this is actually the first clone of DNA from the environment, a big chunk of DNA coming from SAR86. Until this year, SAR86, gamma protobacterium, was uncultured until this year. So we had to look at a chunk of the DNA in order to understand or try to predict what those microbes are doing, because we cannot culture them. So first of all, we have on this chunk of DNA the ribosomal RNA. So this is the way we detect the candidate for sequencing. And then on it was a small open reading frame that was coding for something like bacteriorhodopsin. I'll show you bacteriorhodopsins in a minute, but just look at all the open reading frame. All of them are, or most of them are, housekeeping genes. So based on housekeeping genes, we cannot say what those microbes are doing. So we're looking for those unique open reading frames that can tell us something unique about those lineages. So this open reading frame was coding for something that did look similar. It was somewhat similar to bacteriorhodopsins. So bacteriorhodopsins were found in the 70s in Heloarchaea, and those are actually proton pumps. But our discovery was actually the first microbial rhodopsin in an abundant bacteria. So no, not archaea, but bacteria from the marine environment, and those SAR86 are actually more or less 50% of microbes in the photic zone, and they had those rhodopsins. I guess Massimo will talk after me more about how those rhodopsins are pumping protons. But what was important for us is that we were able to express, and remember, this is from 2000. So at 2000, we only knew very few microbial rhodopsins. We knew the bacterial rhodopsins, those are proton pumps, the helorhodopsins, chloride pumps, and then different sensory rhodopsins. By function, they are actually doing what visual rhodopsins are doing. And what's nice with the rhodopsins from bacteria is that you can easily express them in E. coli and study. So here you can see E. coli expressing rhodopsins, and the chromophore for those rhodopsins is all trans retinal. So you can see the nice color, and then we can really study them and see how they function. So when we discovered this, so this is the protorhodopsin from SAR86, and at that time we knew quite a bit about the bacteriorhodopsins from Heloarchaea. So bacteriorhodopsins discovered in haloarchaea in the 70s. So those are pumping protons outside using light energy, and then the proton ATPase are doing ATP from this gradient. And then those archaea do have other, as I said, they have the halorhodopsins, chloride pumps, and then different sensory rhodopsins. How abundant are those? And I think this is an important slide because you can estimate, at least in the more or less different marine environments, how many rhodopsins you have in the different genomes compared to photosynthesis. So here we have different stations. Okay, so you can see coastal stations, reefs, open ocean, and others. And in each station, using metagenomics, we estimate how many genes for photosynthesis, oxygenic, anoxygenic, and then rhodopsins. And look at the color code. You can really appreciate that in most stations, we have more rhodopsins, or at least more rhodopsin genes. And it's the genes. And nowadays, we do know that about 70% of microbes in the photic zone actually or do have the capacity to express rhodopsins and basically harvest light energy using rhodopsins. So 2 slides about briefly different proton pumps, rhodopsin different proton pumps. So here is bacteriorhodopsin on the left. So they absorb orange light. And then protorhodopsins next to them. There are some that can absorb blue light, some green light, and there are more. And here you can see the different discoveries in the last, I would say, 10 years of here. We have now rhodopsins that are pumping sodium, light-driven sodium pumps, and different chloride pumps that are different from halorhodopsins. So this was a brief introduction, and now to different discoveries from my lab. So here we are actually doing samplings in Lake Kinneret, Sea of Galilee. And here we are using just E. coli that would become pigmented in order to find the positive clones. So here you can see 96 well played with different clones, different DNA from the environment, and one of them gave color once we add retinal to the libraries. And this turned out to be a very interesting rhodopsis. You can appreciate this deep purple color here. And luckily to us, because we were looking for pigmented E. coli, we actually found a new family of rhodopsins. So here in this slide, you can see that we have the microbial rhodopsins on the left. We call them type 1 rhodopsins. Next to it, the visual rhodopsins, type 2 rhodopsins. And here, this is the new family. If you look at the sequence identity compared to type 1s, this is less than 15% identity on the protein level, really the limit to see anything. And what's nice or interesting is that the orientation in the membrane is completely opposite to type 1s and type 2s, so completely different. Here I want to mention, and I'm not sure how many people in the audience know, but actually some rhodopsins, some microbial rhodopsins that are proton pumps, they have their own light harvesting complexes or the ability to have another chromophore that would transfer energy to the retinal molecule in the middle of the protein. So this is an old discovery from already 2005. This is a different type of proton pump. So it's not protorhodopsin, but very similar. We call them xanthorodopsins. So here you can see the protein inside you can see the retinal. And what's nice is that from outside, this protein would have a carotene. In this case, salinic xanthine. And actually, this salinic xanthine can absorb in blue and transfer the energy to the retinal that otherwise would absorb in green. So actually, those rhodopsins have the capacity to get energy from blue and green light at the same time. I'm returning to this slide again, telling you that about 70% of marine microbes do have rhodopsins. And for a long time, since actually 2005, in the beginning with Janusz Lani and then my lab, we were trying, we gave it a lot of effort to try to see if the marine ones would have also antennas, the marine rhodopsins, to have antennas. And actually, until 2022, we failed. We couldn't say that those rhodopsins do have an antenna. So actually, we needed to think out of the box in order to answer whether those 70% has or have the capability to harvest light using light harvesting complexes. So in order to find those antennas, the approach was very simple. In this case, we started from Lake Kinneret using metagenomics. We found different rhodopsins, but at the same time, we extracted different carotenes from the same environment. Okay, and then we combined everything, and we were looking for rhodopsins that actually can bind carotene. And here you can see the result. I'm just showing this only more or less one or two slides, that actually those rhodopsins, protorhodopsins, but also environmental xanthorodopsins, they can bind, in the first case we tested, lutein and zeaxanthin. So those carotenes are used also for photosynthesis, but actually those rhodopsins are using those as antennas. So lutein and zeaxanthin, they absorb in blue. And then at that paper, we were able to show that we can actually see about 40% increase in light harvesting when we add those antennas. So those light harvesting complexes are really helping those rhodopsins. And the distribution, you can see here different stations, so different marine stations, also different lakes. And if I'm looking at the color code of the different stations, if you can see a yellow light, it does mean rhodopsins that have the capability to bind antennas. And in blue are those without. So we can see that mostly in lakes, more than 50% would have the capacity to bind antennas. In the marine environment, it's about 25 to 30% at different environments. So it does mean that most, I would say, rhodopsins in the environment have the ability to bind antennas. I'm left with 10 minutes to discuss and have questions. Just a brief thank you. A lot of people are involved in this research, and of course, we get money from different funds, and I'm open to questions. Nancy,
Thank you.
Thank you, Nancy. I'll skip. I want to see the faces. Okay, so I'm stop sharing. Okay.
Thank you, Oded. That was very, very exciting. Questions?
Hi, this is Anthony Bernini from Georgia Tech, and I was wondering, can you hear me?
Yeah.
Okay.
You go to this lectern and
Okay.
into the
So you can see, there we go.
yeah.
Here we go.
Can't see. So anybody have a question come up?
There we go. So I was wondering, you're talking about these rhodopsins that have the antenna that constitutes a single molecule shoved up against the rhodopsin itself. Do you think there's a reason that we don't seem to find rhodopsins with fancier antennas like some chlorophyll things have, with another protein with multiple pigments pushed up against it?
Yeah. So until now, we are looking for those. So every extract that we do for carotenes, we get also different chlorophylls there. But until now, no binding. But we are open. So I would say that what we did was a bit limited. So only 2 publications now on those different rhodopsins from the environment. And we recently published about Asgard rhodopsins. And in that case, the diversity of the carotenes that they bind is much larger. You have, I would say, exotic carotenes that do bind and transfer energy. We are also looking for rhodopsins that perhaps can bind 2 antennas at the same time. But this is the future. We don't know yet. Yeah.
Great.
Looking for your talk.
Hello, yes, I'm Barry Bruce. I have a few questions. Are any of these new rhodopsins trimeric, like bacteria rhodopsin, or are they all monomeric?
No. I guess Massimo is better, would answer this better, but most of them are, let's see, hexamers? Massimo, am I wrong?
Oh, no, it's okay.
He said, no, it's okay. I don't know. It's okay that you had the wrong answer.
Yeah.
What
So
is
usually
the trimers? Okay.
No,
Okay.
white trimers. No. Proto-rhodopsins are using, let's say, sometimes they are working as 5 and sometimes as a complex of 6. Okay?
Okay.
Yeah, the heliorhodopsins are working as dimers. This is another family.
Or bacteria rhodopsin, but anyway.
Okay. One more question is on the inverted topology forms. You know, and you're in Israel, so you know that there's this interesting protein EMRE by Shimon Schuldeiner. You know about this protein? Has an inverted topology. Have you ever looked to see, is it both topologies or just one topology that's inverted?
You talk about the heliorhodopsins. So with heliorhodopsins, all the heliorhodopsins we tested, and we tested several, all of them had the inverted topology. Yeah.
Oded, we have a question in the chat. Can you comment on why anoxygenic RC1 organisms seem so underabundant?
Could you repeat the question, please?
Okay, I'll repeat it. Hi, the question is, can you comment on why anoxygenic RC1 organisms seem so overabundant? Or underabundant.
They're under. Yeah. I mean, if you look at the publications on anoxygenic photosynthesis in the marine environment, this would go only to 10% out of regular photosynthesis. So I guess most of the electron flow of photosynthesis in the marine environment is oxygenic photosynthesis. And anoxygenic photosynthesis is only 10% or less. Is the question why? Or
The question is why RC1 types are so I think the answer is because they're all anaerobes. But
could be, I don't know.
one last question.
Great talk. Thank you. Sorry, this is about a bit out of my field. You talked about the marine environment, and then when you talk about terrestrial, you talk about it being in lakes. Are these only found in organisms within aquatic environments, or can you imagine you'll find them on rocks or in soil or anything that would be considered more solid and terrestrial? And if no, then why?
Yeah, but the answer is yes, yes, yes. You see rhodopsins in rocks, or, you can go 1 millimeter deep, you still see rhodopsins. Ice, what else do you want? They are everywhere. Whenever you have light, then you see rhodopsins that are pumping protons and producing energy. Yeah. So soil, the top part of the soil, rocks, ice, yeah, everywhere.
Okay, one last very quick question.
Hi, I'm Allie. I have a really quick question. I'm wondering if you are finding any differences in the photocycle kinetics between the bacterial and the proterodopsins.
Again, this is a better answer question to Massimo, but photocycles of the proton pumps, I would say, perhaps Massimo will kill me for this, but this is more or less the same. It's a fast photocycle. If you need to pump protons or other ions, you need a very fast photocycle. In the case of those, this is, I would say, 10 to 20 milliseconds. And then the sensory rhodopsins, you have photocycles of 200 milliseconds. You need longer photocycles because you need to transfer the movement of the protein to another transducer. But then, there is this entire field of people that are working on the different cycles. And then every bit of a change of the photocycle, then they make a story out of it. But for me, doing microbial ecology, I'm just looking whether it's a fast or slow photocycle. And for the pumps, you need a fast photocycle.
Thank you.
You're welcome.
Lindsay tells me we still have a little bit of time. I have a question again. For those organisms that use the carotenoids like lutein and so on, do they make those themselves in the cell, or do they pick them up from the environment and incorporate them?
So there was a recent publication, not from my lab, and this is, I would say, of interest here too, because it's a bacterium that is doing anoxygenic photosynthesis, producing, in this case, zeaxanthin, using this as for the light harvesting complex. And whenever there are different switches, turn into a rhodopsin phototrophy kind, and using the same carotenoid for the protorhodopsin type of phototrophy. So some of them can produce, some of them somehow, we don't know how those carotenoids are entering the cells, because most of them would have an outer membrane that prevent the...
Right.
Yeah, but I would say about 25% do produce their own carotenes.
One other question. Can any of these organisms live photoautotrophically, or do they all live photoheterotrophically, the ones that
with rhodopsins, this is a very good question. So far, no evidence. I know that there are some ongoing manuscripts. People try to describe rhodopsin-based CO2 fixation. Yeah, so there are some, I would say, at the very early stages of people reporting that yes.
So it's a possibility. Yeah.
It is a possibility. Yes.
Yeah, that's relevant, I think, in the context of this conference.
Yeah.
Do we have time for one more?
Just a quick follow-up. How about, hi, this is Kevin Redding, Arizona State. Follow-up question, how about any organism that can grow purely phototrophically? Forget about carbon fixation, but just getting its energy solely from light using rhodopsin. Is that known? Because every example I know of, it's supplementary ATP.
Yeah, I would say this, perhaps shooting in my leg, but if we talk about rhodopsin-based light harvesting, this is really inefficient compared to regular photosynthesis. So in our estimations, because perhaps also Tony can comment on this after that, but we think that they're adding to something like only 3 to 5% more energy. But most of the environments I'm talking about are oligotrophic. So even gaining 3 to 5% more energy, and this was shown in some cases, this is a game changer. So again, rhodopsins would add only small amount of energy to the cells, only 3 to 5% more energy.
Thank you so much, Oded. Let's
Thank you, Oded.
Thank our
Great
speaker
talk.
again. Thank you. Thank you. Great.
So our next speaker is also on rhodopsins, Massimo Olivucci. Massimo?
Yes. Okay. Thank you very much for having me, and of course I'm very grateful to the organizing committee for this opportunity to address you with some research that is being carried out in my lab. Please don't expect any information on the rhodopsin photocycles because I'm not an expert. There is other people that are experts, as you will realize very soon from my talk. So Oded has been talking about the use of methods, which are the functional metagenomic methods, which are informing us on the diversity of rhodopsins and also, led to the discovery of new rhodopsins. My job is different. My job is to show you how the methods of so-called computational quantum chemistry can be used to perhaps study the adaptation of rhodopsin to illumination condition, perhaps also illumination conditions in a planetary environment. Okay. So at this point, we know a lot about rhodopsin. We know that it's an extremely simple system when compared to the photosystems, just one protein and one pigment in the minimal level. And we also know that the key player here is a molecule which is called a protonated sheaf base of retinol, and it is 6 conjugated double bonds forming, let's say, an electronic structure which is highly polarizable. Okay, but it's also a cation. Okay, so it has a positive charge, and this positive charge is stabilized by usually a negative charge, which is called the counter ion of the protein itself. The other thing that we have learned is that in order to function, to trigger the so-called photocycle, is the photon that is absorbed is actually trigger a photochemical reaction. So this is going to be actually a, let's say, specific, let's say, selective double bond isomerization, which is in microbial rhodopsin is usually the isomerization between the double bond in position 13, 14. So remember that, okay? So now here, my assumption is that the efficiency of these particular systems and basically their adaptation, by efficiency I mean adaptation to illumination conditions, is a function of the so-called primary event in proton pumping. Primary event means simply 2 things. And the first thing is that the particular rhodopsin has to be able to absorb a specific light wavelength that is in the environment. That is the first constraint. The second constraint is obviously that hopefully the isomerization has to be highly efficient isomerization. In this term, we call it quantum efficiency of the isomerization itself. So basically, if one wants to investigate the efficiency of the pumping of a proton, it has to mainly focus on these two particular events, the absorption of a specific wavelength on one side and the quantum efficiency of the isomerization on the other side. So now I'm going to tell you that nowadays the method of computational quantum chemistry have been advanced at a stage good enough to be able to address this adaptation problem. By addressing, I mean study, of course, the mechanism which leads to high efficiency, but also possibly predict high efficiency in maybe unknown systems, even variants. And of course, if you want to use the methods of computational quantum chemistry, you need necessarily to build a computer model of the protein itself. And this computer model has to be based on a particular kind of quantum chemistry, which is called a multi-configurational quantum chemistry, okay? So let's say that the model you end up with is basically seen in this slide, and it is called a multi-scale, or other people call it the QM/MM models. Basically, the models is composed by two subsystems. First of all, is an atomistic models, and these two subsystems are the one that is reacting to light, which in this case is obviously the retinal, in green here. And this is treated at the multi-configurational quantum chemical level because you have to simulate the motion not only of the nuclei, but also of the electrons. So the electrons have to be there because they are going to be photoexcited, of course. And the rest of the protein instead is represented in a classical way through basically charges and van der Waals interaction, which, however, impose a field on the first subsystem, the quantum mechanical subsystem. So basically the, let's say, electronic wave function, the electronic structure of the retinal is feeding the field of the environment, which is simulated at a lower computational cost with classical physics, let's say. Okay, so the problem with these methods that, by the way, were originally proposed by Ariel Warshel, which because of these methods, the invention of these methods won the Nobel Prize for Chemistry in 2013, is that they are time-consuming to build, extremely difficult, technically difficult to build, and also error-prone. So it's very, very easy because of the number of variables you have to take into account when building this model. And because of that, it's very difficult to use them systematically in biology, because you know that in biology, usually you want to compare many different types of proteins of the same family and discover comparatively their differences. This is because about 8 years ago, we decided to basically build a machine, okay? A new machine, which is producing this type of model automatically. Automatically means that those models are not produced by humans, but are produced by a computer program, basically, okay? And you can see here the flow chart of this program. Basically, you input a structure, which could be an X-ray crystallographic structure, it could be a cryo-EM structure, it could be an homology model structure, and automatically the program is producing this QM/MM model. And the QM/MM model can be used then to study the function. And the function in our case is the so-called primary event in proton pumping, so the one that I've been defining a moment ago. Just to let you know which kind of results and advancement in trying to understand the adaptation you can get, I'll show you, let's say, a collection of results using this technology, this new technology. And as you can see, these are basically the absorption maxima, the lambda max computed in terms of excitation energy or vertical excitation energy, computed for 32 different rhodopsins coming from 20 different organisms. Not only proton pumping rhodopsin, of course, but we have visual pigments, vertebrate, invertebrate, and of course a number of microbial rhodopsin. And we compare the observed lambda max with the computed lambda max. Okay, so you can see that basically there is an error bar because it's definitely is used 3-4 kilocalories more in excitation energy, but we can study the correlation, and the correlation is what is most important, I think, as far as I know, for biological studies. Okay, so to see the changes, to predict or see the changes. The other thing that is quite amazing here, you can see from this diagram that rhodopsin really span a very large, absorption range, because they go from 690 nanometers absorption. There are rhodopsin they are absorbing the red substantially, up to the UV, basically, and close to the UV. So consider that this is the same chromophore, basically the same chromophore. So the big deal here, from the scientific point of view, I would say, is to understand how the protein environment is actually tuning the properties of the same chromophore, exactly the same chromophore in a sense. Okay, so that is the target that was mainly the subject of our research during the recent year, and I show you some results of that. So, for instance, one thing we wanted to understand why, I don't know, in spite of the same identical chromophore, you have a very different absorption bands for two different rhodopsin, like bacteriorhodopsin, which is, the canonical proton pump, and neorhodopsin, which is the rhodopsin which is absorbing 690 nanometers, so very, very in the red. So you can see that the model that we have is also reproducing the absorption band, not only the excitation energy. We can compute the entire absorption band, and you see a comparison between the experimental absorption band and the computed absorption band there. And so you want to wonder, what does the protein environment do to span the change the lambda max of such a large amount? And it turned out that electrostatic effects are probably the primarily important to this thing that we call color tuning. Basically, technically it's called color tuning here. And it operates the operating environment by exploiting the different electronic structure of the ground state and the excited state of the molecule. Okay. So, for instance, you see that the ground state has an electronic structure such that the famous positive charge in the retinal chromophore is localized basically near the nitrogen here of the Schiffer base moiety. But the excited state has a charge which is translocated along the conjugated change, basically on the other side. There is basically a motion, is actually electron transfer here, but it's an intramolecular electron transfer, a motion of the electron density of a 0.5 electron, which is moving from this region into this region, and therefore the positive charge is translocating in the opposite direction, going here. Now, think about now the electrostatic field that the environment, the protein, will project on the retinal chromophore. If this electrostatic field is such which is stabilizing the positive charge in this region, then you will have a blue shift, because you stabilize the ground state with respect to the excited state. But if the electrostatic field is destabilizing the positive charge in this region, then it means that you will have a red shift, because of course this gap will decrease by destabilizing the ground state with respect to the excited state. So it's all a matter of the electrostatic field generated by the protein environment to modulate the lambda max. So basically to get a large gap or a small gap between the ground state and the, of course, in the excited state. So this was obviously analyzed using this QMMM model I was telling you about, because they can produce an image, a detailed image of the electrostatic field produced by the protein environment. You see here the protein environment of the two types of rhodopsin surrounding the retinal chromophore. And you can see here in the case of bacteriorhodopsin, you have a negative electrostatic potential in this region where the positive charge is in the ground state, but not in the excited state, where the positive charge will be here. While in the neo-rhodopsin, which is a much redshifted with respect to the bacterial rhodopsin, you see the electrostatic potential here is almost positive. So that's the protein environment is able to tune in a fine way the electrostatic field in such a way to regulate the lambda max. Now, I wanted to pass now the second type of prediction, which is instead the efficiency of the photoisomerization. We have been talking about the primary event in proton pumping. That's a primary event also in vision, okay, in our eye. And most of the studies here have been done using the visual pigment, but from the chemical point of view, the system is the same. It's a rhodopsin, a single protein, and a single chromophore. And now I'm going to explain to you how you can use this model to compute the quantum efficiency of the photoisomerization. So what you do, you construct your model, and you then generate a Boltzmann distribution at the wanted temperature. So if you know the temperature where, I don't know, this organism is actually living. And this Boltzmann distribution is substantially represented by a collection of snapshots, a dynamical snapshot of how this model is moving to represent the Boltzmann distribution. And then what you do, you project a collection of these molecules on the electronically excited state. At this point, they are not in equilibrium anymore, and the molecules start to move as a function of time. So basically, you are simulating an ultra-fast photochemical reaction by propagating a trajectory on the electronically excited state. And this, sooner or later, spontaneously, this model will reach a point of so-called conical intersection where the system is abandoning the excited state and is decaying to the ground state, okay? So this is called a non-adiabatic transition technically. So then you follow each trajectory. Some trajectory will reach the product side. That will be a successful isomerization. But some trajectory will also go back to the reactant side, and that will be an unsuccessful, non-reactive photoisomerization. And of course, the quantum efficiency of the photoisomerization itself can be computed by the fraction of trajectories that reach the product with respect to the total number of trajectory. Consider that the total number of trajectory represent the total number of photons that have been actually absorbed by the molecule, okay? So that's, of course, is a statistical quantity. We are doing a statistical prediction here. And the largest is the number of the trajectory, the smallest is going to be the error that you make in this prediction. So we are usually able nowadays to run about 500 trajectories for this type of system, but sometimes you can also use LETA to converge to a stable value of the quantum efficiency. At this point, since I've been talking about trajectory, I want to show you what kind of information you can get by looking at this trajectory. And you will see that from the point of view of the physics of the photoisomerization of the photochemical reactions, it's going to be very sophisticated type of information. And so here you see the representation of the so-called pi electron density. This is a real calculation, it's not a picture. And all these basically red things here represent basically the double bonds, okay? So it's called pi bonds in chemistry, of course. And they are all conjugated, as you can see here. Now I'm going to start the movie. There's a movie that represents a real calculated trajectory. Pay attention to this part because this is the double bond that is going to isomerize, since this is a model of our own visual pigment. Actually, the bovine rhodopsin, I mean, in the sense that the one used for vision, okay? And this means that it is this bond, the 11-12 double bond, which is going to isomerize spontaneously. Okay, so this is how the reaction is going on. So you see that light is able to break the bond. The bond is now broken. You are in the excited state now. And the molecule is starting to isomerize. You see that basically there is a torsion about this bond. And then now you decay to the ground state. You are in this point and you are decaying to the ground state, and the bond has to be reconstructed, but it is reconstructed, of course, with a different stereochemistry. It's now a trans bond, it's not a cis bond anymore. So that's the kind of information that you get not only at the level of nuclear motion, but at the level of electronic motion. So that could be very important considering the effect of the electrostatic field on this type of reactions. Okay. Now, this is basically the result of the calculation of the quantum efficiency for bovine rhodopsin for the visual pigment, but would be the same for bacterial rhodopsin or proton pumping rhodopsin. You can see here that we are monitoring as a function of time 200 femtoseconds, because this is an ultra-fast photochemical reaction which is completed in 200 femtoseconds. You can see that you start from the Boltzmann distribution. This is the Boltzmann distribution, the value of the torsional angle, which are not a single value because of the thermal energy, of course. And then, of course, this is the evolution of the value of this torsional angle as a function of time. And then you see all this red circle and blue circle. Red circle means the trajectory that are successful, trajectory that reach the product. Blue circles are trajectory that do not reach the problem, that go back to the original configuration. So you have a number of products, in this case trans, and a number of failed photoisomerization traces. That's why you can compute now the quantum efficiency, and the computed quantum efficiency in this case is 0.71%. And the observed quantum efficiency, there are very, very few observed quantum efficiency because these are very, let's say, difficult measurement to do, is 67%. So there is a kind of 4% discrepancy between the computed quantum efficiency and the observed quantum efficiency. But the good news is now you can do some very sophisticated kind of study, because you can use the same model also to construct ancestor rhodopsins, okay? So you can see here something that we did recently, but also in collaboration with an evolutionary biologist, UT, Belinda Chang. And you can see that you can basically predict the sequence of the rhodopsin of a dinosaur, an extinct organism, obviously, and using our technology to build up the model of this pigment that existed more than 200 million years ago. And then you can see if it is working the same way, it is the same efficiency that modern rhodopsin have. Okay, so now again, because of the systematic, the possibility of computing systematically this type of models in a consistent way, you can make comparative biological studies, okay, in the computer, not necessarily in the laboratory, either to understand or to predict. And you can see here a comparison between the result of the quantum efficiency calculation. I dream that one day will be possible also to apply this technology to the range of rhodopsin that Oded is discovering basically every day. But you can see, you can compare, for instance, the photoisomerization quantum efficiency and also the dynamics, of course, in a dinosaur to the one of a modern spider, the jumping spider, to the one of a squid, which is living in, deep in the ocean, basically in the dark, and to the one of a fish. And for each of these, you can compute of course, quantum efficiency, 66 per 10, 76, 55, and 70. Most interesting here to this crowd, you can do that also for, of course, a material thing. But then things are starting to be extremely more complicated. So we have several examples of this type of calculation for microbial rhodopsin. This is the case of an rhodopsin that was mentioned by Oded and Oded discovered. And you can see that now the photoisomerization is kind of more messy in this type of organisms, something that has been also verified experimentally at a certain level. And you can see that in this case, the retinal is trying to isomerize on two adjacent double bonds simultaneously. Some trajectory are going to try to isomerize along the 13-14. Other trajectory are going to isomerize about the 11-12. So it's like this trajectory tells that the retinal is kind of confused. It doesn't know if it wants to be the retinal of microbial rhodopsin, type 1 rhodopsin, or if it wants to be the retinal of animal rhodopsin, type 2 rhodopsin, okay? And then in the end, of course, you have a jump and hop from the electronically excited state to the ground state, and only the product of the microbial rhodopsin apparently are produced. The isomerization about the alternative double bond is not completed. Okay, that's it. I wanted to stop here, and this is basically the take on conclusion. But you may be able to see it. I don't know if the slides are provided. I want just to advertise that, for those of you that are interested in rhodopsin, there will be an international conference on retinal protein in Siena, in my second institution, and you are all invited. This is my group in Italy, in Siena, the picture of several people that contributed to these studies, and this instead is a smaller group at Bowling Green State University, where I'm spending a quarter of my time. Thank you so much for your attention.
Thank you, Chris.
If you can go to lecture to ask questions and the people on the lecture, you better, so you don't mind.
Thanks so much. That was great. So when you were comparing the spectrum of the bacterial rhodopsin versus neorhodopsin, is that how you say that? You addressed the difference in lambda max, but not the pretty extreme difference in broadening and peak broadening between the two. Do you have any idea about the peak broadening, the molecular basis of that?
Yes, of course, the cavity, the protein cavity, has two kind of effects. I've been talking and highlighting the electrostatic effects, but there is the second effect with the steric effect, okay? So, and then, of course, it's all a matter of Boltzmann distribution, okay? When you compute the Boltzmann distribution in the ground state, you are actually seeing how the, retinal region and the cavity is vibrating. So, and if it is, let's say, a little bit looser, you're getting a broader spectrum. If it is a more constrained environment, you're going to get the thinner spectrum. So that can be also rationalized and engineered in principle.
Hi, great talk. Very quick question.
Yes.
On your tuning slide,
Yeah.
if you could very quickly go there. I think it was slide number 7 or something.
Okay, let me see if I can get it. Color tuning, you mean?
Yeah, perhaps going here. There is clicker.
Okay, but
Here, just click there.
okay.
There you go.
Yeah. So I don't think I got that from your explanation. If you did, I'm sorry if I missed it. On the second spectrum, what is the shoulder? That we are seeing?
Yeah. That's good. Okay, so basically this is something that we miss, but it's a second electronic transition. Okay, it's S0 to S2. Okay, which is S2 should be forbidden, but actually this molecule is not symmetric. Which is the symmetry is broken, and so there is a little bit of a mixture between allowed transition and forbidden transition. So you get a shoulder with a much lower oscillator strength, much lower intensity.
Okay, and
it can be simulated also.
Again, that was my follow-up. Okay, thank you.
So it's not a vibrational subnet.
There are cases which
in
are
chlorophyll, you always see a shoulder like that. That's a vibrational.
It could be
vibronic
transition, but in this very case, is a second because it's not far.
Yeah.
So S2 state is very close to the S1 state. In fact, in certain animal rhodopsin, they are also interacting.
Thank you very much. So about the evaluation of quantum efficiency, look like a very heavy calculation, but I'm interested in how you generate the configuration because considering the time scale on the animation, maybe not MD. So what kind of
Yeah.
in a way?
Yeah. So the question is about the time required to do this type of calculation. Of course, to generate the initial conditions, so to simulate the Boltzmann distribution, that's not extremely time consuming because you can use a mixture of standard molecular dynamics, all based on force fields, molecular mechanics, no quantum calculation, and a little bit of quantum calculations. And then you reproduce the absorption band. From that point on, to run trajectories is much more expensive because then you have to use multi-configuration quantum chemistry, which is different. But to give you an idea, while 10 years ago, the trajectory that we have seen would be months of calculation, nowadays, in a week, we can do basically the entire sampling. Depends on how many processors you have, because you can run a single trajectory on different cores of processors. Okay, so it's weeks for generating this type of evaluation. Weeks of computer.
One final question.
Very quick question. Electrostatic effect of this color tuning is that protein environment is very important. Is water involved as well?
Yes. So in the electrostatic effect, I include everything that represents the environment of the chromophore, including waters.
Thank you, Massimo. A very nice talk. So, we'll now finish out this session with 4 short talks. And the first one will be given by Anthony Bernetti. Anthony?
All right. Hello, everybody. I can do this. It'll be fine. We can just keep going at a good rate. Yeah. So, so I'm Anthony Bernetti from Georgia Tech, and I will be talking about rhodopsins and chlorophyll with respect to priority effects inhibiting the repeated evolution of phototrophy. So what got me interested in this topic was just a big question, not just a photosynthetic question, just are major evolutionary innovations and transitions predictable, or are they super unpredictable and contingent? And different people have come to wildly different conclusions with respect to this. Some people thinking that everything in the history of Earth is just incredibly unpredictable, and we just are here, so we see what we see because of anthropic selection effects. Other people look at what happened and say, oh, well, this makes sense, this makes sense, this makes sense. It's just a nice set of things that'll obviously happen. And what's weird is they're all looking at the same thing and coming to these different conclusions. And what this tells me is that it's more vibes than systematic. So I decided to at least try to be systematic about this because it's interesting. Some major innovations keep happening over and over and over again, like multicellularity. Some form of it has happened 50 times, particularly complicated and interesting, like 6 times. This keeps happening. Apparently, it's accessible. But some are what you call evolutionary singularities, things that happened exactly once and they changed the world. Things like the origin of translation in the genetic code, that's a really big deal. Origin of eukaryotes, metabolic pathways like nitrogen fixation, or of interest to photosynthesis types, oxygenic photosynthesis within the spread of photosynthesis. But the thing about a singularity is it's real difficult to study. There's only one. You can't compare anything. And there's multiple reasons that they could be singular. You could imagine attrition of multiple origins such that only one remains. You could imagine them just being difficult to invent, so you have anthropic selection effects. Or what I think is an interesting possibility is ecological interactions across time creating what are called priority effects. This is a term from ecology where something comes in and colonizes a region and prevents something else from colonizing it. You can also talk about evolutionary priority effects, where something evolves and prevents the evolution of anything too similar to itself. So, and I decided to study these priority effects using phototrophy, the innovation that drives almost all biomass on Earth, and it's incredibly important for reasons I do not need to get into in great detail for this audience. And I just think I think it's the second most important thing after the ribosome in the history of life on Earth for obvious reasons. But what's interesting, phototrophy was invented twice, not once and not a bunch of times, in the form of the retinalophototrophs, rhodopsin, and the chlorophototrophs using chlorophyll. That's weird. 2 is a really weird number. I've talked to physicists, and I don't remember who first said this, but I've had multiple physicists tell me there are 3 natural numbers, 0, 1, and lots. That you either have some reason that there's 0 or 1 of something, or it just keeps happening. So let's take a look at these 2. They are really different. I don't need to go into a lot of background because of what's just happened, but you've got redox chemistry versus an isomerizing proton pump. They're complex and huge versus simple and small. They are primarily vertically transmitted versus rampant horizontal transfer. I didn't write this down, but also like mostly specialists versus mostly generalists. And they conserve a lot of the energy or a small amount of the energy. The thing is, we can compare them. Because there's 2, we can compare them, unlike true singularities. We can look at their evolutionary history, we can look at their environments, their needs. What I'm going to focus on, though, is their physiology. And I'm focusing in on 2 particular variables, their flux per unit protein that they get out of light, and their yield per unit resource that they get out of light. And so I've done an analytical model, basically, of their theoretical optimum performance in different environments. And so this is, here you've got random anoxygenic type 2 reaction center and a rhodopsin next to each other, color-coded, the same as the chunks of my model. Basically, you've got a central engine that actually does the catalysis, which has a yield per cycle and the maximum rate it can run, a mass, and an amount of light it itself can gather. And you couple it to a variable amount of an antenna that has an absorption per unit mass, which is recycled and photodegraded with constants. All these constants I get from enormous amounts of literature. And you apply certain different amounts of light to it. And all of this is detailed in a paper that got finally published a few weeks ago. So, and so basically you can examine the 2 variables that I think are really important, the flux per unit protein and the yield per unit resource, across different light levels. And basically at each light level, I find the optimum antenna mass X, and then count in terms of maximizing these variables. And I look at these variables across different light intensities. And so here, you've got light intensity across a lot of orders of magnitude up to about direct sunlight at the equator. And you can see that at low light intensity, they all are more efficient per unit light. And at high light intensity, they all have higher flux per unit protein. But at low light intensity, chlorophyll wins hands down in both variables, actually, even though this is, they're both low here, but it's much, much higher. At high light intensity, the rhodopsin wins hands down on both variables. And you can actually graph these 2 variables against each other because this is a trade-off. There's a fundamental thermodynamic trade-off between flux and yield in a lot of biochemical reactions. And so you graph this trade-off, each of them has a trade-off, and they cross. And together, they represent this Pareto front of optimal systems, that everything's going to evolve up to this front. And together, each of them is each of them represents sort of an optimal solution in certain environments. And actually, that crossover point, it actually corresponds nicely to some data about where you find chlorophyll versus retinal in the ocean at different light levels. At the high light levels above that crossover point, you've got lots of retinal, and below it's where you get lots of chlorophyll. And so there's this really nice niche partitioning between them where they are dividing the world between them in ways I'll gloss over a little bit for now. And I think they're unlikely to divide the niche so nicely if the properties were random. And this is all about an evolutionary priority effect, preventing the evolution of anything too similar to themselves. And so like you would have this deal where each of them is able to evolve up and reach this Pareto front, and other ones are unable to. And glossing over a few things to make it to something. I think that this also means that retinalophototrophy probably had to come first, because the retinalophototrophy, it only needs an energized lipid membrane. The chlorophototrophie needs a complex electron transport chain. But so like this is both about what each of them need and what their prerequisites are, but also chlorophototrophie can fix carbon. Retinalophototrophy cannot. If either of these filled its entire curve, you could picture one of them preventing the other. But chlorophyll, even if it's very bad at fixing carbon, it can bust through that priority effect because it can do something the other one cannot. And I think this has implications for the purple Earth hypothesis that people talk about. I don't think that it actually suggests that. I think this is actually not a terribly useful idea for astrobiological purposes. You can talk to me about that after this. So thanks for inviting me. Any questions?
Time for one quick question. Barry, have you got?
In your energy investment, did you consider the fact that amino acids take 2 GTP molecules per peptide bond, so that the bigger the protein, the more upfront biosynthetic costs you have?
When I was calculating the flux, one of the variables is flux per unit protein. So just rather than talk about the cost of the protein in terms of ATP, I just calculate like protons pumped per second per kilodalton as one of the variables, so that takes that into account.
Okay, thank you, Anthony. Very interesting. Our next speaker is Kevin Redding from ASU, and he's going to talk about heliobacteria, a subject near and dear to my heart.
All right. Thanks, Bob. And thanks to Bob, Nancy, and Chris for inviting me to give this talk. I also want to thank the Department of Energy for funding this work over the last 15 years, and I'll recognize the people who did the work as I go. So heliobacteria, what are they? I think of them as the world's simplest phototroph. They're the only member of the firmicutes, so they have no outer membrane. They can't make oxygen, nor can they tolerate it. They can fix nitrogen, but they don't fix CO2, so they're not photosynthetic, they're phototrophic, but they can get 100% of their energy from light. They have a homodimeric type 1 reaction center. Bacterial G is unique pigment. It's the closest to chlorophyll A, as you've seen from Dan's talk. And they have no peripheral antenna. They only phototroph like that. So they're the answer to the question, what would you get if you took anaerobic soil organism like Clostridia and gave it the genes for phototrophy? And here are those genes. There's a gene cluster in all the heliobacteria. You have the pigment synthesis genes, the photochemical reaction center, cytochrome BC, and the cytochrome in between. That's part of this light-driven cyclic electron flow pathway, which probably seems familiar to some of you. Here's the reaction center oxidizing cytochrome C, using light-driven electron transfer to reduce ferredoxin. In the cytosol, you go through the ferredoxin to the nicotinamide pools to complex 1, which reduces quinone, which is in turn oxidized by cytochrome BC complex, passing the electrons to cytochrome C, and along the way pumping enough protons, about 8, to make 2 ATP. At least that's what we think, for every 2 electrons you cycle around. So basically light's just driving this little carousel of electrons going round and around, pumping protons and making ATP, and that's phototrophy. Here is what is encoded by that gene cluster. So we think everything else was pre-existing in the original firmicute bacteria. Here's the structure of the reaction center that was determined by Chris back when he was a grad student working with Raymond Froma. I was going to say he looks younger, but you look the same. I'm not jealous at all. So anyway, in young Chris's beautiful structure here, the PSHA protein forms this homodimers. So it's a perfectly C2 symmetric structure. It's really the simplest, 2 copies of one polypeptide. There is an extra polypeptide, this PSHX, but we knocked out the gene for that, and it's not really required. When we look at the electron transport chain, so here's the special pair of bacteria chlorophyll G we call P800 because it absorbed 800 nanometers, in analogy of P700 to photosystem 1. The photosystem 1 cofactors are in gray. And you can see the arrangement's very similar. Here's the FX4 and sulfur cluster. The A0 is a hydroxychlorophyll A. It's the only chlorophyll A in the organism. It's right here at the primary electron acceptor position. But notice what's missing. There's no quinone like in photosystem 1. So at the time we thought, well, because these are little
further down, and this is a little bit closer. Maybe it's close enough the electron can hop across. There was, however, a molecule that we couldn't explain at the time. There was a long isoprenyl tail that ended not in a quinone, but a tetrahedral group interacting with an arginine. Thought this was probably a phosphate, so this would be the main quinone is minoquinone 9. Here would be nonaprenyl phosphate. We know that these are involved in a cycle to build cell walls, so it's available in the membrane. So maybe it's incorporated in the protein. And sure enough, when we extract pigments, do LC mass spec, we find the right mass. Here's compared to a standard nonaprenyl phosphate, and we get the expected stoichiometry of 2 per reaction center. 75% isoprenyl 9, so that explains the stoichiometry. I asked Chris to say, okay, keep the isoprenyl tail, but replace the phosphate with the quinone. Where would it be? And it's here to the side of A0, not really in a good position to be an intermediate electron transfer. Abhishek took that structure and then did molecular dynamics for about half a microsecond, and it kind of moves away from the arginine and nestled between the serine and tyrosine. And you notice it's right behind the A0 chlorophyll, okay, very close to it. So the prediction is that it's so close, if it were there, it would always take the electron from A0. A number of years ago, we did an experiment with helibacterial membranes, and if we expose them to light just for a few seconds, the minoquinone went down and the minoquinol came up. So it looks like the reaction center can reduce quinone to quinol, which is unusual for a type 1 reaction center, right? Okay, so Adam did a bunch of molecular dynamic simulations with different cofactors and different redox states, providing the frames that were the grist of the mill for all the quantum mechanical calculations that he and Dmitry Matrashov did. So what we're looking at, here's the simplified picture. Charge separation occurs in about 20 picoseconds, Charge separation occurs about 20 picoseconds to the chlorophyll A. So this is what I'm called A0, analogy to PS1. And then subsequently in about 600, 750 picoseconds to FX. But if the quinone were there, the rate to that is predicted to be about 4 times faster. So it would pretty much always reduce the quinone. So here's our model with isoprenyl phosphates doing. It's serving as an endogenous inhibitor to prevent quinone from being there and taking the electron, because this long pathway gives you 8 protons pumped, 2 ATPs. But we know that the stoichiometry is such that the complex 1 is probably limiting it in the high light, and then it would cause the electrons to back up. And then once that happens, you can't get electrons out of FX. That might drive a conformational change to kick it out, allow the minoquinone come in, probably get only a fraction of the reaction centers, and then they can start doing the short cycle, which gives you half as many protons. So this is why you don't want to do it normally, okay? Only in conditions where the ferredoxin pool is reduced. Okay, so I'm going to finish up one story in the next 2 minutes. Looking at the A0 to FX, which I thought was going to be very simplistic because it's been measured before, but Dimitri's group didn't really want to do it. I understood finally, here's the Marcus equation for electron transfer rates. And this is the coupling constant as a function of distance. It falls off exponentially. Here's the A0 to minoquinone right there in a nice short pop. And that gives you that rapid time. But for A0 to FX, they actually had to extend the calculations to get, it's never been, electron transfer had never been calculated for a hop that long. It's about 18 angstroms. And you get about micro electron volt range for coupling, which gives you an optimal transfer rate about 6 microseconds or so. That's about 3 to 4 orders of magnitude slower than what we measured. Dimitri said that's impossible. There must be an intermediate that it's going to, that you're missing. So we checked to see, is there an intermediate? Basically, the answer is no. We can remove the quinone. Here's A0 getting reduced, and then in this recovery is the electron transfer, electron leaving A0. You can remove all the quinone, doesn't matter. You can mutate the only aromatic group, a tyrosine, in between to methionine, doesn't really change anything. So essentially, we've done all these things. I'm just going to fast forward through it. We've changed the FX cluster. We made mutations nearby of certain residues that can change the potential, and you do affect the rate. So if you mutate near FX, you affect the rate of electron leaving A0. So it is a direct electron transfer. But look at this. We shift the potential down 100 millivolts, and we only see a 2-fold effect on rate. And this mirrors, almost said, mirrors what we've seen with other mutations. Very small effects. It's very robust. And here's the thing. I'm not going to go through all these methods, but what we really need to affect, we can change it delta G all you want, but you really need to affect the coupling. And the only way we could do it was to
consider super exchange using the protein as a virtual bridge, using basically the higher energy orbitals as a bridge between. And that can give us a coupling increase of about 87, which is proportional to square. That gives us the 3 to 4 orders of magnitude. Here's one of the major pathways from A0 to FX, hopping there to that tyrosine, but to the beta methylene group, along the polypeptide to, the cysteine over here, and then to the iron. Remember, this is a virtual hop. These are not actual electron transfers. And so I'm going to summarize all this. Photochemistry matters. You're always starting out with one electron. You got to combine the 2 electrons if you're going to make a bond. Does it happen in the reaction center? Does it happen later? And what are the potentials of your acceptors that are going to determine how complicated your pathway is and how much biological energy you can extract from it? And if you're going to have rapid long-distance electron transfer, you may need to use a super exchange mechanism. The bad news is that it's hard for me to test. And if you like to look for sequence motifs, you're not going to find them because it's based on structure, not sequence. But the good news about that is it may be easy to engineer and doesn't require polypeptides, so a form of life that uses a different polymer could still use this. And I'll stop there.
Okay, maybe time for one quick question. Chris. Mr. Helio.
Thanks, Kevin.
Professor Helio.
So one thing I'm wondering about is, you say there's not a very clear way to test this, but there has to be a good amount of sequence homology to photosystem 1. Could you change just a...
Yeah, I didn't have time to go into it, but you can do experimental, computational experiments and knock out individual. And except for the cysteine, which is a ligand to FX, none of them have much of an effect.
So that tyrosine doesn't?
So that tyrosine, even though it's part of the bridge, we changed it to methionine, didn't care because it's the beta methylene carbon is jumping it, well, virtually jumping into. Sorry, this is confusing. Welcome to quantum mechanics.
Okay, on that note, maybe we should move on. So our next speaker is Colin Gates, and I'll let Colin give this title.
Thank you. All right, so I will try not to be as long-winded as I usually am. I'm going to be talking about what happens if you put things other than the normal manganese and calcium in the water oxidizing complex of photosystem 2. Sorry, oxygen evolving complex. I will try to remember to use that term, and it's stuck. Okay, good. Right, so photosystem 2 is, of course, the only water oxidase that nature has evolved at this point, and it has this catalyst that Junko and Usha have given such a great introduction to already yesterday. So I'm not going to belabor this, but okay, that's just not working. Right, so it's possible to substitute several of the inorganic cofactors in photosystem 2. I am not going to talk about what you can do with the bicarbonate or the chloride in photosystem 2 today, but I am going to talk about the things that are directly in the oxygen evolving complex. So your calcium and your manganese, you can successfully replace, as Junko said, the calcium with strontium. And we have been able to replace the manganese with cobalt. And the way that this is possible is if you have extremely highly purified photosystem 2. So microcrystals that have been generated via a series of repeated recrystallizations, then since you have such well-controlled photosystem 2, you can regulate what the available acceptors are for it and get amazing efficiency of operation of the water oxidizing complex. So in this case, with optimal acceptors, you're able to get up to 50 periods of oscillations. For comparison, in a living system, the best I've ever seen is about 17, and usually if you're lucky, you'll get 3 or 4. So the water oxidizing complex in these crystals can be removed fairly easily by addition of a reducing agent. We've used hydrazine or hydroxylamine in this case. I'm going to be talking about what happens if you use hydroxylamine. And then just by adding, and so this is going to cause the native manganese to be reduced to manganese 2, and it will readily leave if you just wash your crystals, you can wash out all of your manganese pretty easily. So then photosystem 2 is really interesting because even after you've taken out its catalyst, it will reassemble a new one by taking up manganese, calcium, and bicarbonate if you have an adequate light level and available electron acceptor. So we were able to fully deplete manganese from from photosystem 2, and then as I said, you can reconstitute it. And if you reconstitute with manganese, you will get partial recovery. And this will still produce active photosystem 2 with the new water oxidizing complex in it. But if you add cobalt, over time, you are going to wind up with much more cobalt than you would have with manganese, and it just keeps going and going. So obviously, this is far more above what you would expect if there's a stoichiometry of 4 manganese per photosystem 2. So we wind up with more than twice as much as you would expect. So what's going on here? Well, we actually get some active photosystem 2 that is capable of doing a period 4 cycle with cobalt in it. And again, that is not manganese contamination. We did ICP-OES. There's enough the contamination level is enough to explain about 2% of what we actually got out of that. But the overall activity is only superficially the same. You still get the period 4 oscillation, but there is this massive decay feature as compared to what you see with manganese. So it also has a higher resting S2 population, and so it's going to decay. It's going to be decaying 2.5 times as fast, and whereas these centers with manganese in them are going to remain active over time. For hours and hours of continued operation using flash oximetry here. With cobalt, you are going to get these tapering off, and eventually, in a much shorter time, you will wind up with all of your cobalt substituted photosystem 2 is going to be dead. It's not going to be evolving oxygen anymore. So I'll come back to that, but essentially, what it looks like it's doing is many of these photosystem 2s are just producing cobalt-3 oxide. Now, there's something. Now, the only other somewhat successful substitution of the manganese in photosystem 2 has been an attempt by Boris Simon and Mike Siebert in 2015 to put iron in photosystem 2, and in this case, you actually get the native water oxidizing complex reacts with the iron 2, which causes reduction of some of the manganese. You lose half the manganese in your photosystem 2, and then iron replaces it. And you wind up with this partially manganese, partially iron cluster that is capable of producing peroxide, but does not go all the way to water to full water oxidation. Wow. Okay, that should say oxygen. I should not edit my slides at 1 in the morning. Yeah. Right. So that's just to briefly touch on calcium. Calcium can only be substituted by strontium. Effectively, you can get it partially through the cycle with cadmium, which is similar in size. However, strontium is the only successful substitution that you get, and we have a structure of this from now over a decade ago, and this causes some reorganization of the oxygen evolving complex. We grew Thermosinococcus, from which that crystal structure was obtained, with whole cell substitution of strontium, and if you look at, and so this has been done for decades at this point, you get very similar oscillations in the activity of the water oxidizing complex with strontium as compared to calcium. So this, I will note though, this is a thermophilic organism, that you grow at fairly low light intensity. However, so strontium is not beneficial normally to the flash oxygen activity. It is beneficial, however, if you add quinone. So there's something else going on the acceptor side. I'm going to just skip that in the interest of time. Strontium is actually beneficial to the rate of oxygen evolution under native light conditions at higher temperatures, though. So you will get more oxygen on a per photosystem 2 basis, but only under very high temperatures. Now, strontium is actually detrimental to the operation of the water oxidizing complex. It's lengthening specific transition times. The S1, S2, and the S3, S4, S0 transitions are slower than the native organism. This is probably because strontium favors a certain configuration of the water oxidizing complex. This more closed as opposed to the open configuration. It's larger. That's probably what's going on there. And Yulia Pushkar summarized this fairly recently. Yeah, okay, I'll wrap it up. Yeah, so overall, strontium, you can survive strontium substitution in the water oxidizing complex. Cobalt would really not be a good thing to have in a water oxidizing an alien water oxidizing complex. Iron is not a good enough oxidant on its own to go all the way to oxygen evolution. And simply, manganese is more abundant than cobalt is. Iron's more abundant than manganese, but yeah, just doesn't get there as far as oxidants go. Yeah, sorry for taking so long. This was done in part while I was a grad student at Rutgers and working with Petrofrome at ASU.
Time for one quick question.
Hi, thanks. Great talk. You mentioned these hybrid iron-containing catalysts. What about potential contamination? I believe you that basically accounted for 2% of the activity, but could you also have hybrids with your like with manganese and the cobalt, for instance? And also, did you do ICP-MS on the grown cultures of your organism?
On the grown cultures with strontium? No, I have not. I don't have ICP OES data to show for that. As far as what was the first part?
Basically making chimera crystals. I don't know what to call that even.
Yeah.
Cobalt and manganese.
That's on the agenda. The big difficulty there is it's really, really hard to get crystals clean enough for this to be particularly viable. More frequently, we just get the overproduction of that strange cobalt oxide that doesn't evolve oxygen.
Okay, thanks.
Okay, thank you very much. Our last speaker of this session, thank you, is Trevor R. Trevor?
That's the arrows, and this is the pointer.
Hi, I'm Trevor. I am here to talk to you about efficient light harvesting. To introduce myself, I am currently a postdoc at UC Santa Barbara, but in a couple months I will be an assistant professor at the University of Rhode Island. And kind of all of the things I'm going to talk about today were done in partnership with Nathan Gaber at UCR, who unfortunately could not be here. Both Nathan and I work on kind of the dynamics of quantum systems and light matter interaction, mostly in kind of artificial systems. But we have kind of longstanding interest in what I sometimes call biological optoelectronics. That is, of course, photosynthesis. And the thing that kind of interests us and our flavor of physicist is the concept of kind of power conversion efficiency, which is how you kind of robustly convert your instant light into useful energy, especially given that photosynthesis has to deal with kind of rapidly varying light conditions. I like this kind of video to illustrate it because if you consider the perspective of a single plant cell or photosystem within it, it's undergoing varying conditions kind of all the time as it blows in the wind. We heard a lot about Bob's estimate of 10 photons per system. Well, not all the time. And to my flavor of physicist, we are fascinated by leaves blowing in the wind for kind of this reason. And so we wanted to understand just some fairly basic physics of how this works and what implications it might have. And whenever I talk to kind of interdisciplinary contexts, I have to kind of make a note of how we're going to approach this, because I don't think I need to tell this audience that the photosynthesis is really complicated. But the kind of advantage of trying to understand the very basic physics of something is that you can be highly reductionist. We can kind of take the complexity and just try and pick out one piece of physics we want to understand. And that's what we're going to do. We're going to do what is often in physics called a toy model, which is only sort of trying to sweep a lot of the details under the rug and capture one little piece of physics that we can play with. In the business, this is sometimes called solving a spherical cow, based on the old joke that a physicist would be kind of useless on a farm because we can only solve problems for spherical cows in a vacuum. But the reason that we actually do this and is that what we're searching for is something that is exactly solvable, that we can kind of play with convenient mathematics. And since we're only kind of trying to understand one aspect of the physics, it doesn't need to capture everything in order to be useful. All of that was kind of a long-winded way of saying that I'm going to take a really intricate system and boil it down to just a few things. So I'm going to say that photosynthesis tends to involve two pigments like chlorophyll A and chlorophyll B that are absorbing fairly narrow bands compared to the solar spectrum. So you have a solar spectrum. There's some distribution of photons as a function of wavelength, and my pigment molecules are picking out some narrow pieces of that. And as they are kind of absorbing energy, that energy is going into some sort of charge transfer network to become an output. But I'm going to just kind of stick with this level. We've heard a lot about what goes on inside that network in various systems. Our goal here is more to kind of solve this basic system using some mathematics borrowed from network theory and see if it can have any implications for photosynthesis. So now I'm going to be a little bit more precise about what I mean by power conversion efficiency. If I want to kind of efficiently be converting my instant light into useful energy in conditions that may change, what I want to do is I want to match the energy flux coming into my antenna complex to some sort of demand that I'm capable of dealing with. And so the other sort of corollary to that is that if I want to do this well, I want to sort of not have more or less energy coming in than I can handle. And that means I want to kind of suppress fluctuations. So the first condition we kind of write down as a matching model, where you consider the sort of input power of one input channel. That is sort of script piece of A is the power that might come in through a channel, and you multiply that by a probability, and the probability of absorbing a photon and having it enter your system. And then you match that to, I'm trying to match that to some output power omega. And this is just a basic expected value. And if I'm considering two pigments, then I'm adding in a second channel called channel B, again with probability B, power B, into this sort of matching condition is my first point. But the reason that I'm kind of constructing this in a probabilistic way is that as we're thinking about power coming into the system, we're thinking about a statistical distribution of energy coming in. I'm kind of illustrating that numerically in a simple case where it just ends up being Gaussian. This sort of second condition power conversion is saying that I want to avoid being overpowered or underpowered, which is to say that I need to, if I want to worry about the scale of the variance of this distribution. And so in order to kind of reduce fluctuations, I want to reduce this variance. And the reason that I kind of set up the model in this way is because if we use this mathematics, then there's a theorem that lets me write down what that variance is. And so I can just write down an expression for the variance of, say, a single channel system. But something interesting happens when we consider having a second input channel. And that is, if you write down the variance of the two-channel system is equal to the variance of the single-channel system times some factor which is less than 1 if you set this ordering up. That is to say that by using two channels as opposed to one, I can reduce the variance coming through my system. Which is, again, the thing that I wanted to do to improve my power conversion efficiency. Now, at this point, this is kind of very abstract mathematical fact, and I want to see how it can apply to photosynthesis. And it can under certain conditions. And so what we can do is we can solve for the conditions that reduce variance and improve power conversion. My old PI Nathan gave this sort of a more poetic name. He likes to call it quieting a noisy antenna, sort of reducing the sort of noise in your process. And it comes down to, again, I'm sort of approximating the system as being couple narrow bands that are being absorbed, and then I get a choice of where I can put these in the spectrum, and that will affect the quantities on the previous slide. And let me actually, if I consider what happens as I move these around, A and B around, that will let me solve the system. And I'll spare you kind of all the details, but noise reduced when you have two channels that have a specific operable bandwidth with respect to each other. And in particular, that are located where the slope of the spectrum is large, which makes the channels both kind of distinct in their, this sort of reduced variance would not work very well if I were absorbing kind of right at the peak of the spectrum where you might maximize the number of photons, but on where you expect, there is a large slope in the input spectrum. All of this is to say that we can kind of calculate what is the ideal absorption that would quiet a noisy antenna if I had consider a given solar spectrum. For example, if you consider the solar spectrum arriving at Earth, which is this gray line, I can calculate sort of that this set of absorption peaks are good from this perspective. And if I then take this sort of prediction and compare it to what chlorophyll A and B actually do, we see that it sort of corresponds pretty well. You have pigments with the right bandwidth that are absorbing sort of at the right position in the spectrum. And this is saying that the chlorophyll is exhibiting a structure that is kind of improving its efficiency by quieting the noisy antenna. Now, just to be clear about kind of what we did here, we considered a very simplified model. There's a lot more going on in chlorophyll. There's a lot more reasons why chlorophyll is an awesome molecule. And this doesn't sort of take away from any of that, but it can partially explain pigment structure. And it can do so in a way that is very interesting because it's biology agnostic. There's no real sort of biology or biochemical details in this model. It's just based on what is the input spectrum. And that means that we can make some interesting predictions. We can think about, we can work with collaborators who can tell us about bacteria that live, sort of under a canopy of trees where the solar spectrum they see is modulated. As a result, a large amount of it is cut out. And you can run that through the model and see what it predicts and find that a lot of them exhibit forms of chlorophyll that seem to, again, have the same structure. And you can keep going for more examples. For example, green sulfur bacteria that live in the ocean, or there are even more examples I could go through where a lot of photosynthetic structures on Earth seem to exhibit the same basic phenomenology. And that sort of lends itself well to the topic of this workshop of extraterrestrial photosynthesis, because we're making something that is a biologically agnostic prediction. If I just approximate the sun as a black body with some effective temperature, I can run that through the model and get a prediction for what pigments sort of exhibit this physics. And if I change the effective temperature to that of an M dwarf, it will predict a different set of pigments, indicating that if I'm on a planet orbiting an M dwarf and I want to quiet my noisy antenna, then I can give you some sense of what pigments should be, at least some what structure for pigments should do that. This is, I would say, a starting point, but it's constraining a possibility space. And we might want to kind of add more onto that. And in the last few years, about a year, Nathan and I have been connected with some people who have been teaching us more about exoplanets and the origins of photosynthesis. And we hope to have kind of a new publication on extending the noisy antenna model to take a few other physical factors into consideration, coming out soon. And more generally, I want to leave you with the idea that this sort of physical reasoning, these noisy antenna models, can generate sort of predictions with limited information that either we can exactly solve, or perhaps things that we could physical concepts we could realize in a lab and perform experiments on. And that is something that I intend to do at the University of Rhode Island. So with that, let me thank all of the people who contributed to this, particularly Nathan, Richard, and Rank, who taught us about pigments, and our new sort of astronomy collaborators. And let me thank you for your attention. Okay.
If you can come up to the podium, that'll just be it.
Yeah, we're a little short on time, so make it quick. Yeah.
Hi. Great talk. I'm a big fan of this kind of work. I have two questions. The first is, I think I noticed that the peaks that you're predicting are always, I think, the Q bands. Is that correct? Not the Sorey bands of the pigments. And so is there a way to, is there a reason for that? Or is there a way to also include that in the model? I guess what I'm trying to do is make the cow less round and now more shaped. And as a follow-up, because I think it's all connected also to my question, I also noticed that the intensity between peaks is something that your model can't predict yet. Is there a way to maybe develop into that as well? And I'm asking this because for retrievals, all those things matter, not only the position where the peak is. And so it would be incredible to be able to add that.
So I think this is a question that we should talk about offline. I would say that the model at the level that I presented is very generic in the sense of being able to kind of just accommodate, it just predicts a few things. You might start, be able to, as you said, introduce different, make the cow less round and introduce, and you can, and I have some thoughts on that we should talk about.
Okay, we have a whole lineup of questions, so I ask people to keep it very, very quick, and you keep your answers quick too.
Actually, I'm suggesting we maybe take part of the maybe just another working group session to maybe address a lot of the questions that was raised. Because it's a lot.
Go ahead, Roberta.
Yeah, you're optimizing for the antennas that already exist. You're optimizing both the low energy transition and the high energy transition. But if you look at how photosynthesis works, and everything is decaying to the red, to the lowest energy transition, these are the transition that is used for energy transfer. So then I don't understand how you can optimize also for the high energy, the S0, S2, S0, S3, S0, S4, that is what you have in the.
Yeah, so the short answer is that if you have those transitions that are decaying down, they could contribute to the noise if they're kind of in the wrong place, and so you kind of want to consider sort of them in parallel. But I agree with you that things filter down to the lowest energy transition, and that's usually the kind of important one in some sense. But you can also gather more photons by having those higher energy transitions. So if you get those at the right place, it just helps. So.
Okay, I thought that you have calculated also this for the energy transfer, taking into account.
Maybe let's
we can
talk about it a little bit more.
Thank you. For plants, they use both chlorophyll A and chlorophyll B. So using your model is a way to estimate the optimum ratio of chlorophyll A and chlorophyll B in plant leaves.
I suppose you could try and use it for that. More, it's we think about it as optimizing kind of the absorber properties, to the extent that you can tune pigments, but that you could probably, again, if we're talking about this, we can probably do some estimate like that in terms of the abundance ratio.
Yeah.
Yeah.
More questions?
Yeah.
Hi. So my question is just, does the size of the antenna network change what you expect from optimization?
Depends what you mean by size. You mean like physical size or complexity?
Number of interacting pigments.
Yeah, so you can also write down a model for 3 pigments, 4 pigments, 5 pigments if you like. What you'll do is what you'll see if you do that is you run into diminishing returns pretty fast. So if you want to consider having 3 pigments for this effect, you might not get as much improvement if you do that. But again, you're adding another set of molecular machinery, so maybe that's not quite as advantageous. But it's certainly something you can consider.
Okay, obviously sparked a lot of interest. Let's thank Trevor and all the rest of the speakers. So now we're going to have coffee break. Nancy's going to give us our marching order.
Okay, so we had a 15-minute break. It's a little bit shorter now. We'll still come back at the scheduled time at 10:30 for the next keynote. Also, we will be taking a group photo before lunch. Just letting you know that. We will have a breakout reports before the group photo. So, and time for discussion. Thanks.